in fact, these biological systems, they are made from macromolecules which are encoded by the genomes and which interact with each other in order to build this kind of structure. And the particularity of this is that this biological system don't have functions that their part alone don't have. And these parts, as I said, are interacting through molecular interactions to build this system. So network biology is about interactions, cellular functions, and complexity. But in fact, the term networks is often overused by us biologists. A network, in fact, doesn't exist in real. A network is a representation. So we have to realize every time that we speak about networks that in fact, first you have the data that the biologists are producing, which corresponds to observation or measurement, and that this is only by the work of the bioinformatician, which is transforming the data into the network. So the network, as I said, is a representation. And because the network is, in fact, a graph, which is a mathematical abstraction of the network and the data, then that we can apply particular tools and algorithms to this graph in order to get back to the biology. So in fact, when we are studying interaction network, we are learning about the biology by studying the representation of the data. So in the group, we are working since ages now on protein-protein interaction networks, which are also called interactome. So they correspond to interactions in between proteins, which have been deciphered most of the time by large-scale to hybrid screens. And the ones on which we are working are mainly we take them from databases, so they do correspond to the assembly of interactions from different sources. So the consequence of this is that they correspond to, these networks correspond to the set of all possible protein-protein interactions that have been identified between the protein of an organism. And this means that within this network, most of the time, you don't have any spatial temporal information. So here is encoded some biological information. So to get back to the biology, we need, as I said, to study them. To study them, we can develop algorithms because biological networks are modular. So this algorithm will help us to identify these modules. And these modules functionally, so they correspond to the graph into groups of nodes, which are most of the time more linked between themselves than with the rest of the network, and functionally it corresponds to proteins which are involved in the same biological process. And when you study such a big table, you can have some information about some particular processes you are interested in, and you can use this for function prediction, for instance, but you can also perform global graph analysis, which will give you, as I will show you, the idea of the prevalence of some cellular mechanisms. Okay, so in the lab, we are interested since a long time now about multifunctional proteins, and particularly on what has been called Moonlighting proteins. So they are a particular family of multifunctional proteins, and Moonlighting means that these proteins have unrelated function. In fact, moonlighters in the United States means people who have an additional job in addition to their regular one. So in French, it will be like un travail au noir. So these proteins, they have unrelated functions, of course, in the limit of our current knowledge. And here there is one of the main examples of such proteins. So here this is the Aconitase, which is obviously an enzyme of the Krebs cycle, which is catalyzing the transformation of citrate into isocitrate. But when the concentration in iron in the cell is changing, then the protein is changing its function, and then it's becoming a regulator of the translation. And in particular, it's becoming a regulator of the translation, it's binding mRNA, and it's then regulating the translation of proteins, which are important for sequestering of iron in the cell and the iron uptake. We know that this kind of change of functional change is happening because there is a change in structure of a conformational change of the protein. There are also proteins which have moonlighting functions according to the status of the cell, if the cell is in a healthy state or in a pathological state. And in particular, here there is this example of this RAM protein, which is important, is playing a role in the stability of the mitotic spindle in the normal cell, and in the tumor cell is found in the extracellular matrix, where it's able to bind a receptor, which is triggering the transcriptional activation of genes, which are important for the metastatic progression. So then, the question that we ask are the following. Because these proteins, most of the time, they have been found by serendipity, so randomly. We don't know much about them. So we are not able to characterize them as a group. So first, we wanted to try to identify this kind of proteins, because they can be important for several reasons: regulatory, genotype-phenotype relationships, and so on. We would like to know what are the features or the determinant of this functional singularity, and obviously we would like to know how the this functional switch is regulated. So several years ago, we started a network approach to try to get a hand on this protein. And so for this, the reasoning was the following. If moonlighting proteins have unrelated function, because protein perform that function by interacting with other proteins, then we can expect to see this protein interacting with at least two groups of proteins which are involved in two very different functions. So to do this, and because as I said at the beginning, in the interactome you don't have any special temporal information. So this information is probably buried within the network. So first of all, we developed an algorithm able to multi-class proteins within the network, meaning an algorithm which is able to identify overlapping clusters, overlapping functional modules. The idea being that the proteins at the intersection can be multifunctional and maybe moonlighting if the functions are unrelated. To know if the functions are unrelated, then we play with gene ontology annotations, and for this we devised some a method that we have called Pronto, which compute the probability of geoterms association. And based on this probability that I can explain later if you want, in fact, we are able to say if for an organism, these two geoterms are often associated among the annotation of the proteins, meaning that they will describe similar function, whereas if they're only rarely associated among annotation of proteins, then they will describe dissimilar geoterms and dissimilar function. So playing with this, we annotated our module and we got all as a candidate for Moonlighting functions, all the proteins which are found at the intersection of two modules of unrelated functions according to these probabilities. And by doing this, we have identified between 1 to 3% of the proteins of the interacting. Then, because we had this data set, we tried to get a molecular, a signature of these proteins. What do they have as special? So we compare this, our candidate, to the ones which are found at the intersection of the modules, but which are not candidate because we found them involved in the same function, and to and to the rest of the network. So this is the candidate are the blue, the multi clustered non-candidate the yellow, and the network is the black. And here the green. This is the comparison with the hubs of the network. And what we have found is all the features here are found statistically different from the other group. That they have some. So the main thing is that they have a very high degree, and they are even more connected within the network than the hubs. So they are like super hubs. But whereas the hubs are known to be very structurally disordered in order to be able to bind to a lot of different proteins in different conditions, our candidates are not disordered. They are very connected, but not disordered. So what happened there is this. In fact, what we have found, which is very interesting, is that within this, although our candidates are less disordered than the hubs, the disordered region are enriched in ELM that you can see here. It stands for eukaryotic linear motifs, and these are small motifs of 3 to 10 amino acids within the sequence of the proteins, which are mediating protein-protein interactions at low affinity. So, in fact, so here they are. So these are the definition for the short linear motifs. This is the same thing that eukaryotic linear motifs. What is interesting with these small motifs is that they provide the possibility to interact a lot at low affinity. Among them, there is an enrichment of what has been known as switch eukaryotic linear motifs. So they are non-motifs which play the role of functional switch. And in addition, among those, this is the allosteric switch, which are enriched, even though the numbers are very low, but the sample is very low, so it's normal to have such a low p-value, high p-value, sorry. But still, it makes a very nice proposition that maybe the conformational switch, the functional switch in some candidates is due to the presence of these short linear motifs. Then we have put all our candidates into MoonDB. So this is a database of Moonlighting proteins and Extreme Multifunctional Proteins, and which is now cross-reference in a unit. And this is Diogo and Lionel who have done most of this work. So then, as I told you, what appeared to regulate the change, the functional change, is in fact the change in context, as I told you for the Aconitase. So what we know as a reminder is that this change of function can come from a change in conformation and, importantly, in subcellular localization and in interaction partner. So as I explained to you with the HMMR example. But what is striking is that, in fact, these proteins are found in different subcellular compartments, although they don't have anything within their sequence which make us think that they can travel from somehow, in some condition, at some point, from the nuclei to the extracellular matrix. So we went a little bit further in the characterization of the features of our multifunctional proteins, and so we just verified that indeed they are located. So this is the same kind of thing that you've seen before, that indeed they are located in more subcellular location than the rest of the proteins of the network. And so here, this is corroborated by the fact that our EMF protein, extreme multifunctional proteins, are found in other type of data set. The multi-localized proteome of human proteome atlas into the translocatome. The translocatome, this is a database of proteins which are translocated from one compartment to another. This is predictions made on machine learning analysis algorithm. And they are enriched a little bit in plasma membrane protein. But still, as I told you, even as a group, we can see that there is a heavy depletion in addressing signal to go to the membrane or even in transmembrane. We notice also another particularity of our proteins is that they have weird 3'UTR parts in their mRNA. So their 3'UTRs are longer, and they have more 3'UTRs than the rest of the protein of the network. So this is suggesting that maybe they can be regulated through the 3'UTR of the mRNA. So why did we think this? Because in in this work, the group of Christine Meyer in New York City showed that in fact the 3'UTR of mRNA can be involved in the regulation of proteins in membrane in membrane localization and in the formation of protein complexes. So then it works like this. This phenomenon has been described for only one complex experimentally. So what happened is the following. In fact, in the cell, half of the genes have alternative 3'UTRs of different lengths. So in the known case, when you start with the long 3'UTR of the mRNA, and this long 3'UTR is able to bind to an RNA-binding protein, here in green. This protein is recruiting another protein that we call the intermediate protein, which is in fact a protein partner of the protein, which is encoded by the CDS within the mRNA. Yes, okay. And then the nation protein in this condition is translocated to the plasma membrane with the help of the intermediate, which is interacting with small row and rack proteins, which provide the energy for the complex to go to the membrane. What is striking is that when you consider the protein, which has exactly the same sequence, but which is translated from the short, the mRNA, which has the short 3'UTR, then the protein is not found at the membrane anymore, but within the reticulum, because it's not transported, because the complex cannot bind, because this is the 3'UTR is short, doesn't contain the RBP binding site, so the complex doesn't form. So as I said, this has been described only in one piece. And we thought that maybe it can represent, in fact, a way to, for our proteins, which are devoid of addressing signal, to go to the membrane. In addition, if you think about it, when you have several 3'UTRs, and when you have alternative 3'UTRs, then it means that it can help to the formation of different protein complexes, because you can bind to different or none of the RBP, as I explained. And you can have, because you have different UTRs, you can bind to different RBPs, form different complexes, and at the end have different functions. So we wondered if the formation of the 3'UTR complexes could explain the localization and the functional change of our Moonlighting protein. And for this, we devised another network approach. So for this, we started with a network of protein and RNA interactions. We have in two databases the interactions in between a large number of RBPs and 3'UTRs. So we got all this information. It's coming from experiments, of course. We got all the information within the network. Then we map the RBPs onto. So we map the RBPs onto the protein-protein interaction network. Then we map the nation protein onto the protein-protein interaction network. And then we looked for the potential intermediates in between, located in between RBPs and nation protein. And like this, we got complexes. So we got 1,400 EMF, so extreme multifunctional proteins containing complexes, and overall on the whole network, almost 10,000 such complexes. Then we okay, so here are the numbers. So these complexes contain half of our extreme multifunctional proteins, whereas it contains almost 70% of the proteins of the whole interactome. Then we perform randomization in order to check that this number of complexes cannot be obtained by random. And this is what you can see here by comparing these two columns and the empirical p-value here. So we have a higher proportion of extreme multifunctional proteins involved in these complexes, suggesting a possible regulation. And it appears to be a quite prevalent mechanism at the interactome level, since it conserves almost 20% of the proteins. Then we verified that among the proteins which are within the complexes, we are enriched in proteins which have been seen at the membrane, whereas in absence of addressing signal, this is the case. And then we reuse our tool, our statistical tool, which allowed us to define what are the different annotate, the dissimilar or the similar annotation. So at the beginning, so here we use it with not biological process annotation like for identifying multifunctional proteins, but for cellular component geo annotation in order to check if our candidates which are involved in 3'UTR complexes have a tendency to be found located at unrelated or very dissimilar location, dissimilar statistically. And so this is what I just explained. And in fact, this is the case. And this is what you can see on this graph. So particularly the main annotation that we find among the EMF are membrane and nuclei, and as I said, always devoid of addressing signal. So this suggests that we may have a protein trafficking mechanism here between this compartment mediated by the formation of these 3'UTR complexes. So here we wanted to check if these complexes were helping to build different molecular complexes, because these proteins are involved in very different functions. So then we play again with the annotation, and what the idea is that if we wanted to check if, in fact, the proteins, the intermediates, the proteins which are interacting with those and which are involved in the complex are involved in very different processes and very different cellular components for both. And here are the numbers, and in fact 68% of the EMFs which are in the complexes interact with intermediates in modules which are dissimilarly annotated, either at the level of the biological processes or at the level of the cellular component. So this made us even more comfortable proposing that 3'UTR protein complexes provide the diversity of interaction partner and the subcellular locations, which are two prerequisites of extreme multifunctionality. So here just genealogy annotation analysis of the two groups, EMFs and intermediates, in which we can see that we have a lot of membrane related, in red here, annotation, signaling, and location, which make difference trafficking, and the same for the intermediate protein. And very interestingly, we have this RAC protein signal transduction here, which is enriched among the intermediates. And if you remember, I told you that in the only case which is known, the intermediates is interacting with the small GTPases to give the energy to go to the membrane and to change the location. Okay, so 3'UTR protein complexes mediate multifunctional proteins trafficking, maybe, linked to signaling and cell migration processes, which we can see. Okay, so then obviously we verified that our very well-known moonlighting proteins are contained within our complexes, and this is the case. So here so this is the case of the enolase. So the enolase, this is an enzyme of the decolytic pathway, as you know, but it's also going to the membrane and it's found outside the membrane, where it becomes a receptor of the plasminogen, and here is involved in cell migration. Very interestingly, in fact, all the proteins, all the enzymes of the glycolytic pathways have moonlighting functions. We found them all within our complexes. Okay, so as a first conclusion, I told you that we developed network approaches to predict extreme multifunctional proteins in Moonlighting, candidate for Moonlighting, and 3' protein complexes. We found a signature for multifunctionality. We think that they contribute, these complexes, to the subcellular localization and the diversity of interactions necessary to multifunctionality. It can represent 20% of protein interactome in the interaction. Now, very quickly, I don't know how long I have still, I want to tell you about what we have done rapidly on lncRNAs. So, lncRNAs, as you probably know, are known to be involved in the regulation of transcription, and they are also known to be involved in the sequestration or the nucleation of some proteins. And in some cases, they have been described to scaffold protein complexes. So this is what we wanted to ask at the level of the complete protein-RNA network. But the protein, so maybe I have an example. Yeah, yes. Here, so in this case, also this lncRNA scaffolding protein complexes, this is not well known. Here again, most of them have been found by chance, and so we know them. We only know tens of them. And one of the reasons why we don't know such a function for organizing protein complexes is that experimentally, most of the experiment use an RNA step, which allows you to degrade the RNA. So then you get the complex, but if there is an RNA part into the complex, you cannot see it because it was screwed up by the enzyme. So we wanted to know what is the extent to which lncRNA organize protein complexes, and what is the extent of them. So here again, we took a network approach, but as I said, the protein-RNA network is not known. So then only a small part of it is known. So what we have done, we have used a predictor to predict RNA protein interactions. So we use CatRapid, which is an algorithm which have been developed by the group of Gian Gaetano Tartaglia at the CRG in Barcelona. So this is one of our collaborators, and it's based on the physicochemical properties of proteins and RNA. So we have used the algorithm to predict the network, and you can see here 6 million of positive interactions between co-expressed proteins and lncRNA. So just for fun, this is a representation of the size of the network we have identified compared to the protein-protein interaction network we used to work with. So you can see that here the space and the spaces is huge, and with no comparison to the ones we are used to use. So in order to know if these lncRNA are organizing protein complexes, what we have done is to take protein complexes which are known, either because they have been annotated in the literature, in the database, or they have been predicted or identified with a mass spec type of analysis. The network modules, our favorite type of group of proteins. Okay, I'm done. And what we look for is an enrichment in lncRNAs interacting proteins among the complexes and the modules. And like this, we have been able to identify almost 900 lncRNA, which are in fact organizing 1,500 protein groups. So here on this representation, each line is RNA, and each row is complex. And as you can see, there are some lncRNAs which are very promiscuous, and some others which are very specific. And in fact, the data set that we got represent 5 of lncRNA represent 5% of all the ones we have tested, and apparently they are able to organize 50% of the protein complexes we have studied. So the lncRNA scaffolding function seems to be highly prevalent. Some lncRNA, as I said, are general scaffold, whereas some others appear to be specific. And some of the protein groups interact with a lot of lncRNAs, reflecting their function, like the polycomb. So here it's quite interesting what we have done. Here, all the yellow nodes, they are lncRNAs, and all the square in colors, they represent the complexes we have found to interact with those lncRNAs, and they are represented in this network when at least one of the protein composing the complexes have been found to be involved in the same disease than the lncRNA. So this data have been generated completely separately, but in some of the literature you find that the lncRNA is involved, and elsewhere that a certain protein is involved in the same kind of disease. And as you can see, there are a lot of connection in this network, meaning that probably scaffolding by lncRNA could be a dysregulated function in some disease. So, as a conclusion, I showed you the first large-scale approach to detect the scaffolding function of lncRNAs. We have predicted a very large RNA protein interaction network, and 5% of this lncRNA scaffold 50% of the protein groups we have tested, and we are able to propose, based on this work, a disease association mechanism for some of the lncRNA. So all this work has been done by all these people. So what I showed you is mainly the work of the PhD of a great PhD student, Diogo, who is here, who is now a postdoc in Lausanne, and I've participated to Charles devised all the Moonlighting pipeline and the signature of the Moonlighting proteins. Andreas made tremendous work on all this subject as well. Lionel, so is an engineer in the group, and he's a very precious person for us. The student who have done a lot of work and testing ideas. Emmanuel was developed at the beginning of all this story, the algorithm, and all our funding and collaborators. Thank you.